Global Research University for organizing this prestigious conference. And thank you once again for inviting me as a keynote speaker for this prestigious conference. My topic is recent advances in pharmaceutical research and development based on medicinal plants, from traditional knowledge to modern drug discovery. Now, globally, India is the world of pharmacy, and for world globally, the requirement of new molecules through drug discovery by applying various modern research techniques for rheumatoid arthritis or diabetes, or especially vitiligo or skin diseases, leukoderma. The globally all countries still facing the big challenges in this disease domain, and for that the Ayurveda or the isolation of bioactive molecules for the best therapy clinically, it is more significantly better compared to generics or synthetic drug molecule. So many pharmaceutical industries or many research institutions globally still working on the isolation of bioactive molecule from the natural products or medicinal plants. And for modern drug discovery, the bridges the gap between the ancient ethnobotany or ethnopharmacology or compositional biology through bioinformatics or biopharmaceutics or biopharmacotherapeutics. Revolutionary optimization provide unmatched chemical complexity. Nowadays, in India, Himalaya, or in other countries, especially in China, or means a Chinese plant, medicinal plant, or we can say that in Africa, or in Australia, or in US, or anywhere in globally, there is different species of plants and their different genus. So taxonomically, means in the research literatures, the endangered medicinal plants, and so authentication by botanical scientist or botanist, the selection of the medicinal plant is very important at a basic study, research study, so that the legacy, the traditional ethnobotany laid the foundation for the modern pharmacopoeia, means Ayurvedic pharmacopoeia, Indian pharmacopoeia, United States pharmacopoeia. In the official, these are the official books: European pharmacopoeia, British pharmacopoeia, Japanese pharmacopoeia, Canadian pharmacopoeia. So last 40 years, the Food Drug Administration, US, or in Canada, ANDAs, abbreviated new drug substances, or in US, abbreviated new drug applications. These are the regulatory bodies. They approve the drugs, purely synthetic compounds. They are synthesized from the drug, means chemical compounds, synthetics by organic derivatizations, and synthetic with natural pharmacopoeia. Just last 10-15 years back in India, the most of the companies, they discovered the chloroquine synthetic for anti-malaria and with combination artemisinin. So artemisinin is naturally derived from the Artemisia annua plant, medicinal plant. Means what I want to say that the better synergistic effect has been given by in this combination, fixed dose combination chloroquine with artemisinin. Recently, medical practitioners still prescribing in the prescription providing this combination. Means the combination of synthetic with natural pharmacopoeia, means as a bioactive molecule, it gives a better therapeutic effect, better efficacy, better safety, better purity. Means it gives a good standardization of herbal drugs. Natural products are chemically privileged structure optimized by evolution over millions of years. The challenge is no longer efficacy, but isolation, scalability, and delivery. And the phytochemistry, the chemistry of plants, in that stereoisomer, this complexity, the chelation, or that we can say that according to Lee-Chatterton principle, the acid-base concepts means there is a pH, hydrogen concentration, hydroxyl concentration. Chemistry point of view, there is a chains of molecules. In that, the chemistry, the libraries of drugs consistently fail to replicate. There is a challenges in medicinal plant for standardization purpose. It takes more time for extraction, for the isolation, for the characterization into physical characterization, chemical characterizations, for the isolation of the extracts in mortar and in first diagram, mortar and pestle. This is mortar and pestle. It's ancient techniques. It's very old technique to extract. Focus on trial and error. Whole plant use. Then pharmacognosy, the active principal isolation means first after extractions in pharmacognosy 2.0. In the second diagram, the focus on the chemistry means the focus on the target molecule. Because the chemistry is very important for structural activity relationship to elicit a pharmacological action means therapeutic action. In pharmacognosy 3, the shift from single target paradigm to network pharmacology. The drug acting on the various system of the body, cardiovascular or the respiratory or the digestive system, the integration of the transcriptomics and metabolomics in the plant profiling the primary metabolites and secondary metabolites. Primary metabolites are more toxic than secondary metabolites. Genobiotics, mapping synergistic interaction of the whole extract phytochemicals. Moving past reductionisms to map how multiple phytochemicals interact synergistically. Just before some time we had discussed. The one molecule in the phytochemicals, there is a number of fractions of the molecules in various chemical category, whether it is alkaloids or flavonoids or tannins or resins in various phytochemical class. For that, the big challenge is the purity molecule. For that, there is a analytical techniques for the standardization, separation techniques. That is analytical. Across the complex human biological networks, the green chemistry revolutionized the extraction and the isolation. Now we move on analytical techniques, the supercritical fluid or ultrasound assisted or ultrasonication or microwave assisted or soxalate apparatus. All these are for the extractions. These are the advances, these are the recent advances to minimize the time. What is the advantage of these methods, supercritical fluid or ultrasound for the development of the nano size or nanolipid, solid lipid nanoparticles or nanosizer, the incorporation of the nanotubes or nanosensors in the formulation development of the nano emulsion, nano suspensions, recently now nano gel is developing by various pharmaceutical companies, Lupin or other companies also. Microwave assisted, the natural deep eutectic solvents, the extraction yield, processing time, solvent eco-friendliness, and thermolabile preservation also. That is depend on the physiochemical properties of the extract or the active ingredients that present in a extract. And this extraction in various solvent, polarity, non-polarity, or semi-solvent. The natural detecting solvent as act like the plant's own intracellular liquid, successfully isolating secondary bioactive without thermal degradation. That is chemical degradation, thermal. You know, oxidative degradations, thermal degradation, thermal heat, photodegradation, sunlight. Means for stability purpose. Because 6 month data, 12 month data, these these stability studies, why it is required for the shelf life of the drug, shelf life of the drugs of product, expiry date, manufacturing date, expiry date decided on the stability of the drug molecule. You know the syrup, why it is stored in only amber color bottle? Because to avoid rancidification, oxidation, the reactions is it is used as antioxidant. Then the nano carriers, hydrophobic means it is lipophilic, hydrophilic that is water soluble, hydrophobic is a water insoluble. The poor solubility is a big concern because the better solubility it gives a better enhancement of the bioavailability, bioequivalence. Suppose oral administration of the drug means tablet, capsule, oral liquids. Once administered, it reaches to the systemic circulation of the means absorption, distribution, metabolism, excretion, rapid metabolism, first degradation of the potent botanical bioactives, whether it is a curcumin in haldi, quercetin is a flavonoids, quercetin is present in citrus fruit, in orange, curcumin is a haldi, turmeric, the solution engineered nano carriers. These are the techniques, nano carriers, neutralize biological carriers to absorption. Because the once the drug is administered in the tablet form, in the drug product of tablet, it solubilizes in the stomach, gastric juice, pH, in acidic, absorption, then distribution. Because the drug is bind with protein, drug is bind with receptor, then receptor drug receptor complex, then elicit a, once you administer paracetamol, in synthetic or in herbal also, many companies, Dabur or Himalaya or Baidyanath or other many herbal companies manufacturing this formulation in various dosage form, whether it's a tablet, means or solid, or whether it is semi-solid, and these are depends on the route of administration. These solid lipid nanoparticles just discussed earlier. Polymeric micelles, phytosomes, liposomes. These are the various formulation. Result is the enhanced systemic bioavailability. What is the objectives of these nanocarriers? Neutralize the biological barriers to absorption, to better bioavailability, and it gives fast, quick response. You know, compared to tablet, if administered intravenous injections, it gives fast onset of action. Significantly prolong the plasma half-life. Second, the artificial intelligence accelerate it to lead timelines. Nowadays, you know, drug discovers within 1 or 2 years. But in ancient time or before 30 years, 40 years or 25 years back, already we have seen the drug, it was means taken long time to discover the new molecules, because recently the artificial intelligence accelerate hit to lead timelines means through the instruments, the advanced instrument technology for the isolation, for the extraction, for the manufacturing, the automation base, the artificial intelligence, the laboratory information management system, and SAP, or many softwares. The reverse pharmacokinetics, modern clinical outcome back to plant-based targets. In this ethnobotanical text, that is the nanolipid particles, algorithms, rapidly scan, digital botany engine, virtual screening, machine learning, ML, artificial intelligence, Python, machine learning, driven molecular docking. What is molecular docking? The new trends in drug design and drug modeling. The science finder, these softwares, many softwares, that is a pharmacoinformatics or bioinformatics. Predictive modeling, artificial intelligence driven, the absorption, distribution, metabolism, excretion, and toxicity property prediction for the complex phytochemical libraries. Libraries is nothing but discoveries. Deep learning drastically compresses the discovery timeline by applying the natural language processing, historic text, and high screening. HTS, high throughput screening. HTS. Toxicity, bioavailability, binding affinity are predicted in days, not decades. The synthetic biology secures the phytochemical supply chain. And DNA expression in oncology in cancer molecule. For example, vincristine, the isolation of the vincristine vinblastine from Vinca rosea, Catharanthus rosea plant. The gene identification in target plant, that is the uncontrolled ungrowth mass of cell, that is a cancer, that is tumors, benign tumor or malignant tumors, means the apoptosis. Apoptosis means DNA apoptosis means the death of DNA. In the cancer, in the gene identification in the target plant, the sourcing rare low-yield plant metabolites drives over harvesting, global biodiversity loss. Biosynthetic pathway engineering, genetically engineering microbial host, for example, yeast, bacteria, fungi. These are the target in this gene identification in target plant. Microbiologically, genetic engineering, that is a DNA recombinant technology, glycoprotein receptor antagonist. Nowadays, biotechnology, I had already worked with Biocon, Bangalore-based company, the first insulin maker company, Biocon, in Bangalore, to autonomously express the complex plant enzymatic pathways, the outcome, mass producing rare botanical compounds in sterile bioreactors, scaling the chemistry while leaving the native untouched. Means in this slide, the biosynthetic pathway engineering, biological, microbiological host substance, that is yeast or bacterial, the fermentation, and that fermentation through enzymatic pathway, just like alcohol production, enzymatically. Then clinical translation, plant-derived molecules in the pipeline. Already we explained right now, artemisinin derivative is the molecule. This structure of artemisinin, it is a terpenoids, this 7-member ring, chemically, structurally it is designed, this 6-member, means this plant is artemisinin, flower here. And modern breakthrough, these 3D and 4D and 5D, this chemistry, dimensional. There is a stereoisomers, means chemistry, the structural design and modifying these docking studies, new trends in drug designing. Here, novel botanical, these two examples, novel botanical, the immunomodulators, originally traditional malaria remedy. Here, these artemisinins, their combinations. Now, presently the combination is available in market, in medical shop. The combination of artemisinin and chloroquine for the treatment of malaria. The recent advance is optimized by computational biology, the cheminformatics, that is the cheminformatics, repurposing and nano delivery, and recent advance in immunomodulators This artemisinin is different, and this AI-based identified alkaloid complex in 2024-25 recently. They discovered why multi-pathway network pharmacology screening, that is biological screening, the target pipeline actually entering phase 2 and 3 trials targeting neuroinflammation, that is an immunomodulator, Alzheimer's disease. Alzheimer's disease means there is a deficiency of beta amyloid protein. That is a neurodegenerative disease. In the next slide, the advanced metabolomics, same agricultural variability. The target metabolite means there is a biological pathway. The development of or the process of metabolites, primary metabolites convert into secondary metabolites through a ischemic acid pathway. The advanced metabolomics, their separations, analyticals, by applying analytical techniques, HPLC, HPTLC means high performance liquid chromatography, but for analysis of herbal drugs or herbal raw materials. In that, there is millions of fractions of extract in phytochemicals, whether it is alkaloids, tannins, resins, or flavonoids, or glycosides. And in that, there is a phytochemical constituents. Here in the diagram, that these target meta A, B, C, and marker compound is a standard. And these are the samples, metabolite A, B, C. And these fingerprint signatures. These on y-axis the retain, or x-axis the retention time, and y-axis there is a intensity. This intensity means concentration of the sample, and this retention time means the running time. How much time is required to retain the sample after injecting that sample into injection holder or sample holder. This is analytical part. These analytical techniques. That means in the sample there is a number of ingredients or fractions of phytochemicals or chemical compounds in this sample of metabolomics. Their screening has given that this screening is a chemical screening within 3 minutes. Yeah. So, navigating evolving global regulatory framework, that is US FDA for US countries and EMA for Europe countries and WHO guidelines for globally. These are the regulatory guidelines for the registration of the herbal drug products globally as per the regulatory compliance. Then unseen complexities, the friction points of phytopharma, that is pharma is a drug and phyto is a plant, discovery of the active compounds. In vitro and in vivo. In vitro, for example, dissolution studies, and in vivo in the preclinically in the animal studies. The near horizon, the frontier technology in the pharmacognosy, that is control environmental agriculture, then personalized phytotherapy and quantum pharmacology. Synthesis, the integrated eco to pharma pipeline. Now, every countries and every pharmaceutical industries in globally, all countries, their focus on source, discovery, process, formulation, and validation. And they are applying all pharmaceutical industries, all research institutes, their main focus is on cost reduction and quality improvement of the research projects or research work. Key takeaways for the future of phytomedicine, irreplaceable scaffolds means the selections of the plant material, it is authenticated endangered species, and authenticated by the botanist. Then bottlenecks broken. Recently, artificial intelligence and nanotechnology have successfully resolved the historic roadblocks in both compound discovery and systemic delivery. And with the support of pharmacoinformatics or cheminformatics or docking studies softwares, we can easily identify the new drug molecules and their structural elucidation. Then sustainable scale quality control is very important for the standardization of novel drugs. Thank you.